Today, we are delighted to welcome Professor Chad Hewitt. Chad is the current provost at Lincoln University. He's held a range of senior academic and research leadership positions, including professor at Murdoch University, where he established the Murdoch Biodiversity and One Health Research Centre within the Harry Butler Institute, and he served as the Murdoch Defence Research Coordinator. He has also been Dean of Science and Head of the School of Science at the University of Waikato, Pro Vice Chancellor Research at CQU Queensland, and Director of the National Centre for Marine Conservation and Resource Sustainability at the University of Tasmania. Early in his career, Chad served as the Chief Technical Officer, Marine Biosecurity for the New Zealand Government, and as an invasion biologist at CSIRO. Chad's research focuses on human impacts in natural systems, particularly marine environments, and the role of science in shaping management and policy. He has delivered extensive capability and capacity building programs across the Pacific and Indian Oceans. More recently, Chad has been working with philosophy scholars to explore evidence-based policy, examining how scientific knowledge moves through institutions and influences social responsibility and governance. Please join me in welcoming Professor Chad Hewitt to deliver the excellent series address tonight. Thank you.
Thank you, Grant. Start this because first of all, thank you for coming. It's wonderful to see a lovely turnout, especially to listen to a talk about the marine environment. You want me over this one? Okay. And I guess there are a couple of things, and I'll go through them. A few caveats. 71% of the planet is under oceans. About 8% of the planet is in coastal areas, so that shallow part of the ocean that is in the photic zone, about 140 meters or less. And the consequence of that is that's the area where you have the greatest density of life in the oceans. But realistically, 71% of the planet is land underwater, and the seabed, the seafloor, is land. And so I just want you to relate to that. So I'm at a land-based university. I just happen to study stuff that's wet. My observations, very much in Australia as a research provider, as essentially the creator of knowledge, listening to government need and providing advice to them. In contrast, as Grant indicated, I was the CTO Marine Biosecurity in New Zealand, and as a consequence, I was the regulator, but also the architect of the system, which was really about acquiring knowledge that would inform the policies that we undertook. And so it gave me pretty unique insight in terms of the way I was operating at CSIRO, as opposed to how government receives information, how they use that information for decision making, and even more importantly, how they should use information for decision making. And then the last one is really a caveat that I put forward to most people. Bioinvasions is something that we have done in the land-based environment for many, many centuries. We know it as weed management, we know it as pest management, quarantine activity, but of course in the marine environment, it's really only been since about the 1980s that we have paid a significant amount of attention to it. Although the godfather of invasion ecology, Charles Elton, did mention oysters in his seminal book. So there has been a nod to marine invasions. I'd also be remiss if I did not identify the large suite of collaborators and people who have either received the information, paid me for the information, or worked with me to develop the knowledge base that we have, both in the Australian government the New Zealand government, US Coast Guard, as well as a number of research organizations and industries. BHP, early in the Australian piece, was a heavy investor because they were one of the few coastal fleets owned by Australia at that point in time. BHP undertook much of the initial work on ballast water management. So Australia and New Zealand are indeed maritime nations. And in fact, if you look, we have about, well, 67% of the land is underwater in Australia. 96% of the land that belongs to New Zealand is underwater. We have extensive marine estates. And if you look, 85% of the population, as we know, live within 50 kilometers of the coastline. We are an oceanic focused people. And indeed, if you think about it, you can't get away from 50 miles away from the coastline in New Zealand. But the fact that we are on that coastal rim also identifies our connection with Polynesia in terms of how we engage with the ocean, in terms of food, fiber, and livelihoods. Because of those extensive relationships, those extensive exclusive economic zones, we also have a number of transboundary obligations under the UN Convention of the Law of the Sea. Any boundary where we might undertake an activity that would affect a neighboring state is something that we have an obligation to manage, but even more importantly, an obligation to report. And then equally, the geographic isolation that both Australia and New Zealand have evolved under has led to a very significant level of marine endemicity, 30 to 95% by taxa. If you drill even further down, you find unique species at both class and family level. And then, as a maritime nation, our dependency in the modern day on shipping is significant. 99% of all trade that comes to either Australia or New Zealand, much of which is exchanged between the two countries, is in fact by shipping. It comes in sometimes a little slow, but you do receive much of that material. And because we're consumers, it means that much of that material comes in with trade or in, laden. But it also means that for many of those primary industries where we're exporting things like logs, we'd send the logs overseas, but the vessel comes back empty. And as I'll show you later, that means it comes with ballast water. So New Zealand, as we'd be very familiar, has a very significant experience with invasions in terrestrial and freshwater environments. And in fact, much of that was intentional movements. The acclimatization societies of the 1800s, 1900s were very much around making New Zealand feel like home, which meant we brought hundreds, if not thousands, of individuals to try and make sure that they established really well. Did a good job with Gorse, by the way. As maritime nations, of course, it is important to remember that for shipping and trade, it has changed in terms of the geopolitical relationships, but also in terms of the frequency of travel. Australia and New Zealand, very little connection, except directly through Mother England, pre-Opium Wars. With the Opium Wars, there was a bit of trade between the colonies, and then the modern era demonstrates just how connected we are globally. In fact, within 6 months, New Zealand is connected to every part of the world based on individual ship traffic. Now, a couple of biosecurity watershed moments for the marine environment. As I said, we're doing a very good job operating under the Quarantine Act for weeds and pests. We had a very strong focus, especially in primary industries. And then Gymnodinium catenatum, a harmful algal bloom species, was introduced in the 1980s, and it became very prevalent. The bloom-forming algae causes damage to oyster industries, mussel industries, and if humans consume it, we get diuretic shellfish poisoning. So the human morbidity that was caused shut down the East Coast oyster industry for about 6 to 12 months, depending on which bay you were in. And equally, it impacted on the mussel industry here in New Zealand. At the same time, we had two independent invasions of Undaria pinnatifida, the Japanese kelp. It came into the South Island of New Zealand. It was also introduced into Tasmania about the same time. And the Japanese kelp it's a very lush and very beautiful species, can get up to 2 meters long, but it competed with many of our native kelp species. And the consequence of that native competition was it started to push out some, not just the native algal species, but also some of the native fisheries. And then Sabellus valentini, Mediterranean fanworm that gets to about 30 centimeters, it's introduced into Perth in Western Australia, and over the course of the next 5 years, skipped its way from port to port and made it into Port Phillip Bay, where it basically dominated about 60% of the soft bottom flora. The Japanese sea star, Asterias amurensis, and by the way, I should point out that these are the beautiful pictures on the side there. Asterias was found in Hobart, estimated population density about 2 million when it was detected. Doesn't look anything like the native, but people said it was confused with the native. And then within 6 years, it was transferred from Hobart, where it reached these massive populations, into Port Phillip Bay, where it's now present, probably estimated densities at about 40 million. It's a wonderful omnivore, just kind of scoots along the bottom, eats any shellfish that it can find. And when in points of starvation, eats itself. Now, sorry, at 1990, and I guess this is just a little factoid that I want you to keep in mind. Pollard and Hutchings, Australian Museum researchers, recognized 62 non-native species in marine waters in Australia. So that's the baseline that we have to work with, and I'll talk to you through that. Now, as researchers, we have a tendency to be very much invasion ecologists. We think about the invasion process. It's about uptake, transport, inoculation, establishment, and spread. And of course, in the marine environment, uptake, we think about, well, if I'm a ship, I take up ballast. I transport that across the ocean. It takes about 14 days to get from the west coast of North America to New Zealand. And inoculation is I pump that ballast water out. So it is the transport in a fairly rapid time frame of a large volume of water, which includes the plankton, from one location to another, and the discharge into that new location. And then establishment and spread, obviously, you've inoculated, in this instance, you've inoculated a suite of larval species. So they're, after 14 days, they're probably pretty competent and ready to settle on the first hard substrate that they can find. Now, managers really operate in a similar vein, but they use different language. First of all, they're protecting core values, so it is about bioprotection. But secondarily, they're really talking about pre-border or quarantine activities, stop things from getting here, border activities, find it, kill it, or long-term eradication and control. Now, the key there is, what do we call a pest in the marine environment? And that comes back to your idea, what are your core values? What are you trying to protect? And while environment, economic, and social and cultural values are very good starting points, they're insufficient to be able to actually identify the outcome. So both governments have an evidence-based policy approach. They have clearly articulated that the policies, especially in this fight, will be based on good evidence and good knowledge. I'd like to challenge some of that as we go through the talk. Clearly, that evidence needs to be based on information, whether that information is pre-existing, whether that is a responsibility to seek new information, such as through surveys, or evaluation of vectors, or the assessment of the genetics and the taxonomy of the species. Biosecurity management is therefore based on risk and risk assessment, and we know that we've gone down the rabbit hole in terms of how quantitative or qualitative you can be with those elements. Clearly, the management actions that we can have include managing the vector or the transport activity, managing the pathway itself, so the connection between two locations. And yes, I'm using different terminology, Phil. And equally, making decisions about when you respond. So do you respond rapidly? If you don't, have you lost the ability to actually respond? And then lastly, risk communication is absolutely critical, especially if you're going to understand those societal values in economic, environmental, social, and cultural contexts. Those are the elements that will drive decision-making, because those are the votes that you will accrue. And so really that virtuous circle of knowing what values you're protecting, and therefore having the ability to create management outcomes that you can communicate back to the plebiscite, is absolutely critical. So the government responses to those 1980s suite of incursions included a couple of things. In Australia, the establishment of CSIRO's Centre for Research on Introduced Marine Pests occurred in 1994. I was hired to come down and establish the port baseline surveys, understand species and vector risks, and really identify what options we had to do something about it. At the same time, CRC Reef really had a program to investigate tropical invasions, rapidly became consumed by crown of thorns, a native pest species. In New Zealand, NIWA and Cawthron established a marine biosecurity focus, and at that point in time, they were working very closely together. Again, an initial inventory of museums and collections, beginning the process of impact assessment and risk assessment to help inform what we would act against. And then equally, Australia and New Zealand had an ANZAC guideline on in-water cleaning, but both governments, being driven very much by the global stage of ballast water activity, were very focused on ballast water actions. So in 1998, the Ballast Water and Ships Hull Defouling Strategy was released, which identified baseline port surveys and risk-based species assessments. And then the 2000 biodiversity strategy really started the process of bringing it, normalizing it into an aligned biosecurity system across government. The key focus for both were ballast water and that target species list. Now, in Australia, we were very clear. Target species were not the only species of concern. They were examples of species that similar species would be problems. There was a bit of a problem in New Zealand in that they declared unwanted organisms, and therefore it designated which ones we were keeping out. All the others were to be determined. Responsibilities in both countries are very much embedded, or were very much embedded across ministries. And so there was some confusion, who takes lead, where is the point? And given that it was not ensconced in legislation, it became a little bit difficult. Is it DOC in the conservation state? Is it Maritime New Zealand? Is it Ministry of Fisheries? And then lastly, because of a personality, Dennis Patterson in Australian Quarantine Inspection Service, he drove a ballast water management approach, which became a mandatory reporting system early in the 90s. So the first place in the world that required that to occur. Now, those questions were the ones that were identified and very clearly were to support the development of appropriate policy and appropriate framework. Today, I'll talk about these because they're the ones that I could get through. Melding 4 one-hour talks into one, doing 30 years in 30 minutes seemed to be ambitious, so I won't talk to you about the others today. So a global evaluation on the left-hand side in the late 90s really indicated that there was a significant J curve. And this is not an uncommon pattern for invasions. This just demonstrated in the Marina State, we have exactly the same thing. From the 1800s to approximately the present, you saw a number of very significant growth. San Francisco Bay, the most invaded embayment in the world, recognized about 220 species. It's now about 450. So it has continued. And of course, there are a number of salient events that led to many of those invasions. US in general, Pearl Harbor was investigated by the Bishop Museum and again had that beautiful J curve. Australia, similarly, in red, had that increase, exponential increase, and New Zealand, very similar, got up to about 125 species. The UK, interestingly, stopped counting. So that's why it's a nice shallow curve there. More recently, with colleagues around the world, we've actually started in 1965 and just asked the question, what new species have arrived into specific bioregions? And what we've seen is that, again, that increase, especially of Lesseps invasions from the Suez or through the Suez Canal into the Mediterranean, has continued to increase another 500 species since 1965. New Zealand, again, the port surveys, especially around 2000, have substantively increased the new knowledge of that. So that could be a survey effect. But more importantly, we see the effect of that ballast water management plan that had come into effect in the early 90s in Australia. It slowed the rate of invasions. And that is in the face of significant survey, significant effort in terms of looking. And that includes a very strong focus on citizen science in terms of detection and reporting. That global evaluation also identified about 2,700 species globally around the world, 35 phyla, the 95% in the 14 phyla we see here, but more importantly, 55% from chordates, fishes and sea squirts, mollusks, snails and clams, and crustaceans, crabs, lobsters, amphipods, isopods, creepy crawlies, right? And then equally, what we found is that no region of the world had not been invaded, including the polar regions. So Arctic and Antarctic at that point in time, this is now about 10 years old, the Arctic and the Antarctic had also received invasions and were associated, in that case, with tourism traffic in Antarctica, but in the Arctic, it does appear to be oil and gas activity. More importantly, 47% of all invasions appear in the Mediterranean, and at that time, Australia and New Zealand. So it was a real wake-up call for both countries. We are heavily invaded, but more importantly, many of those invaders that are elsewhere aren't here. And so we need to keep our eye out. I talked about the Australian port baseline surveys. We developed a consistent suite of protocols and survey techniques. Yes, that is me, young, and very long hair at that point in time. The surveys looked at multiple habitats. They took quantitative samples. We looked at 39 ports over a 6-year window. That's about 4,500 dives in that window. For each of the team, we had about 12 people on the team. And we collected well and truly over 2 million specimens. And absolutely a fantastic effort. And in fact, those survey protocols have now been employed by 37 nations, including New Zealand, and have been picked up by the IMO as their gold standard in terms of what we use for those assessments. Remember the 62 baseline species in 1990. Australia, at the end of that period, identified 629, order of magnitude increase in the number of species that we recognize as non-native. The port baseline surveys identified 248 introduced and cryptogenic species. Cryptogenic, for some of you who are not familiar, is hidden origin. So we don't know if they're native or not native, but we put them in there so that people understand the risk that they represent. In Australia, it became very apparent it's a temperate problem. At least that's what the data sort of tells us. But if we actually look, the number of non-binomials in the tropics, and I include Perth in that because Perth is the only tropical to polar current on a western margin in the world. It's very, very unique. So the vast majority of specimens that were collected were not named. So it's very difficult to identify, are they native or not? And if you look in comparison, New Zealand now has 703 recognized, introduced, and cryptogenic species, with over 200 as their baseline because they very much on top of their museum collections in the 1990s. So it became a real apparent growth. But more importantly, the surveys that occurred using identical methods, 109 introduced and 319 cryptogenic species detected through those survey techniques. So Australia and New Zealand receive about as many species as we give away. And I mean, it's a beautiful plot, but it's very difficult to interpret. If you look at that part, that's what we give. And that part, that's what we receive. Now, Australia and New Zealand about equal, but there are some places in the world that are extreme donors, and other places in the world that are receivers. And what this gives you an idea about is the relative propensity of regions to contribute species, whether or not that is because of the strength of trade, or whether that's because of the evolutionary drivers of those groups. So if we use the Australian example of increase between 1990 and 2000, we can see that there is a significant underestimate between point A and point B, and equally, that magnitude differs by phylum. Equally, there are some really underrepresented groups. This is a plot looking at the number of marine regions that have residuals relative to the number of species in the phylum. And you can see that brachiopods, lab shells, echinoderms, sea stars, and sea urchins, entoprocs, nodding heads, and platyhelminthes, the flatworms, are underrepresented as invaders in most parts of the world. And then there's the tyranny of the small and the unusual, which is usually associated with small things don't have a biogeography, therefore we just name them whatever existed in the UK, or we don't have taxonomists. And the loss of taxonomists is a significant element of our inability to identify new species, new invaders. Now, risky transport vectors and pathways, it had long believed that ballast water was it, that we had managed biofouling. But an assessment of that suite of species that we collected in Australia identified that both biofouling and ballast water remained active vectors and that there was a very significant crossover. The difference being ballast water transports plankton, the larval species, as opposed to biofouling, which transports adults. Ballast water, when a ship is empty, it bobs around like a cork and it's easy to break. So what we do is we add water into the ballast tanks, into the holds, which makes it sink into the water and gives it trim and stability. It's estimated that 3 to 10 billion tons of ballast water are moving around per annum, mostly delivered into offloading ports. And then that roughly translates to about 7,000 species moving around at any given ton. And if you look at the global fleet, the current fleet's about 110,000 vessels, about 200,000 vessels 10 years ago, pre-GFC, I should say. And those are vessels that are greater than 100 metric tons dead weight. The Ballast Water Convention has now come into force, which took a long time to negotiate. New Zealand was very strong in its stance. It is very much based on a density standard. You have to get the density of plankton down below a certain threshold, whether you do that through open ocean exchange, shifting coastal water into oligotrophic open ocean water, taking up oligotrophic open ocean water and dumping it into port environments, or ballast water treatment technology that meets the density standards is absolutely critical. Biofouling, on the other hand, is every vessel. Anything that gets wet will accumulate species pretty rapidly, even in the open ocean, and is in a variety of places. It's inside your rudder, it's inside your propeller shaft, it's inside your cooling tanks, it's in any area that gets wet, both exposed surfaces and not. It's estimated that about 570 kilometers square of the global fleet is wet, and it's moving constantly, transporting material from one place to the other. And at present, the only management that we have is either periodic dry docking and anti-fouling penning, or in-water cleaning, which is not allowed in New Zealand and Australia. And if you look at New Zealand's linkage in terms of pathways, its connection, both ballast water and biofouling are very similar, but the strengths are different in terms of where that water is coming from when it comes into New Zealand, as opposed to the number of vessels that transport biofouling between locations. And obviously, Australia is a big donor of ballast water to New Zealand. And remember, 657 new species. So why we do biosecurity? And these are just a couple of our lovely invaders. Economic values associated with economic benefit, very clearly. We have shipping and port services, offshore domestic fishing and agriculture, defense, and oil and gas and offshore energy. It's estimated these marine industries are worth about 80 billion a year to Australia, about 3 billion a year to the New Zealand economy. Environmental values such as ecosystem services, again 104 million per annum for Australia, 357 billion per annum for New Zealand. Difference in methodology there, I would make the assumption. And then in social and cultural values, you can see there recreational fishing and tourism, 31 billion per year for Australia, 22 billion per year for New Zealand. So the marine estate has significant value that is worth protecting, but also potentially at risk. We undertook a program with an Endeavour bid looking at what's at stake, but really one piece of this was to identify what New Zealanders held as significant value. And what we found is, using a different suite of lenses, is that a variety of things that you normally wouldn't think of that could be affected by introduced species came out. Whether the beach or the coastal area was maintained and clean, whether they have access, especially during a management response. The challenge of aesthetics, a number of in-water sports, beach activities, and beach water activities, but equally that care for the environment, the naturalness, and the water quality all came forward as being things that could be impacted by a variety of non-native species that were either here or were incipient invaders. And obviously, they differed throughout New Zealand in terms of where those held values were found. Now, really quick touch on impacts. Impacts are hard. Immediately as an ecologist, you drill down to an experiment, 1 square meter plot, you replicate that multiple times, you got the answer. But the challenge that we have is that there are very few activities that occur. We only have so much money to do those detailed analyses. And the consequences for, we undertook an analysis for biofouling species coming into Australia. 71% of the species had no published information. We knew nothing about them. Nobody had studied them. Of the remaining 200 species, the impacts had to be inferred from statements that researchers had made. For macroalgal invasions, we drilled in very deeply. 80% of 300 species had no information available. What's happened is a consequence. This quote was by an Australian policy maker who said, well, if it were important, somebody would have studied it, ignoring the fact that he controlled the purse string. He was the one who could have invested in finding that out. And that disincentive for knowledge acquisition really belies that epistemic responsibility. You have an obligation, if you're undertaking evidence-based policy, to get the information, to acquire it and apply it. The other is that no detected impact does not equal impact, no impact. 97% of non-significant empirical evaluations for algae and crustaceans were deemed to be insufficient power to actually detect impact until there. So we do these experiments, but we don't actually calculate or report the power that we have. And then the last one, the consequence of that is we have a real bias towards a willingness to miss impacts. There's a bias towards innocent until proven guilty, and therefore we make decisions that may not be conservative. They may not be precautionary. And then, of course, we get to the 90s and the 2000s, more importantly, the 2000s. Late 1990s, the pilchard herpes virus resulted in a kill, 2 kills, waves, from Western Australia to New Zealand. And the pilchard, of course, is an underpinning fish for many of our fisheries, but also our seabirds. The consequence of that is still being felt in terms of diminished populations and distributions. The blackstripe mussel, about an inch in length, was introduced into Darwin. We found it early in the invasion. It was still at 30,000 individuals per square meter. 3 months later, and a real cowboy minister who just said, go for it. $2.4 million and a successful eradication occurred in Darwin. Absolutely fantastic. Scared government because 2.4 million just went out the door. The blackstripe mussel could have affected both the multi-million-dollar houses surrounding the marina, but more importantly, the pearl oyster industry in the top end. And then Sabellus balanzani, this Mediterranean fanworm, was introduced here into the port of Littleton. The government decided to act, put 3.6 million against it, but because of the timing and the density, it was unsuccessful. And so it just gives you an idea of the amount of activity, but also the effort that goes into making this happen. The shift in focus, there was clearly a recognition that ballast water and biofouling were equal and that we actually needed to do something. But equally, there was a shift from preventative management only to rapid detection and early response, developing a tool set that would allow us to undertake activity. In both countries, there was a significant tension between national government deciding not to act, and then that just cascades to the local regional council or to the state, where they then have to live with the problem and therefore act over a long term and spend much more money. Equally, early in the Australian approach, we had ABWMAC, which was a public-private research engagement that was replaced with government-government engagement. And similarly, in New Zealand, it was very clearly a government-government discussion between national and industry council authorities. A couple of interesting surprises, which we believe came through recreational vessels or through the East Australian Current floating across with NEMA. Eudistoma, this beautiful sea squirt. Payura, another sea squirt. Baratasquilla, a snapping shrimp, and Membranicoropsis, a bryozoan, all came into the top end of New Zealand and became quite dominant, aspect dominant. The impact of these is difficult to ascertain. Clearly, Eudistoma is a smothering. You saw significant amounts of loss of seagrass in that context, and the same with Payura. So in 2010 and beyond, what we've seen is a significant slowdown. More coordination, more sharing of information across the Tasman. Recreational vessels are now acknowledged. It's not just the commercial vessels, it's also the small guys, and especially the sailors. Risk evaluations that rely on a suite of information requires that global network and information sharing. And then those impacts on values really need to be further investigated. But how you do that with single species as opposed to generic. And then efficiencies are now being driven by eDNA and citizen science, with some control technologies being trialed, like the response to the Clearpa invasion in Northlands. One of the fears, if you look older invasions versus younger invasions, you can see that older invasions have a much broader distribution. Younger invasions have time to grow. And so the anticipation is coastwise expansion will occur. Equally, if you look at some of the big invaders, some of them actually have only been introduced one place, which means that the opportunity for that expansion, for that new invasion, is quite significant. And then coastal urbanization. As we fear sea level rise, we will harden the coastline more. And as you can see, anthropogenic habitat favors non-natives versus native habitat. And as a consequence, we will anticipate more invaders will be welcomed to our hardened coastlines. And then more recently, obviously, climate-related range shifts are something that is on the horizon. In fact, the intentional transfer of species from more equatorial regions towards the poles to ensure that they survive is something that is also transporting other non-native species with them. Grant mentioned the evidence-based policy work, and I just wanted to end with this. Shortly after I arrived here, we spent they spent 5 months. Grant gave them 3. And we spent a lot of time, philosophers, natural scientists, a political scientist, a lawyer, a journalist, a couple of politicians, actually having a discussion about the epistemology of evidence-based policy and what are the obligations both on the researcher, but also on the decision maker in terms of obtaining that. And I point this out. ZIT is a fantastic opportunity. The University of Bielefeld, funded by the German government, DFG, provide the opportunity for residential fellowships, and twice a year they provide funding calls. And I would highly encourage people to think about that. Take a look at the website, because it's not all about policy and philosophy. Each of you could easily identify something that would give you the opportunity. And the best part about it is they pay for the long-term fellowships, salary, travel, and accommodation. And that accommodation is on site and for families. So a fantastic opportunity. So thank you.
Yeah, Chad, thank you very much for that. Opportunity for some questions now.
I have so many questions. Can I ask three?
Perhaps just start with one, and then we can divide opportunity for some others.
Maybe a question with the bias towards innocent until proven guilty. So what's the percentage of the natural invasion in the current in your study group? Are there any control group of the island country less traveled by the boat, but still have the natural invasion? I feel like I'm not sure if the right term, like a eugenics engineering in terms of ocean animal evolution.
Yeah.
Human molecular.
So biological introductions are human-mediated transports of species, but there are a number of species that get transported by natural means, either flotsam, current-driven transport. Many of those new invaders at the top end, we believe, may have come over naturally on the currents. But the challenge that we have, most of the species that we've identified that are new look like they have been introduced via human mechanisms, agriculture, vessels, shipping, recreational vessels, intentional movements. And more importantly, they're found in ports and marinas. They're not found elsewhere on the open coastline, which kind of gives you a big challenge is the triangulation. The way to identify whether a species is native or not is very forensic and multidisciplinary. You got to look at the genetics, at the evolutionary biology. You have to look at the biogeography, the distribution, both locally and overseas, and the temporal sampling. So yes, there are counterfactuals. And in fact, throughout the Pacific, you can see a number of natural invasions. Introduced species, human-mediated movements seem to be occurring at a much greater rate over much greater distances in shorter time periods.
Thank you.
Yep.
Other questions?
Hello. So I guess one aspect I always find interesting with comparing terrestrial and marine is that terrestrial often interested in more fundamental issues such as invasibility or what makes a species invasive, whereas in the marine area it's very much about quantifying numbers and pathways. And I did wonder whether there is any of that element coming through in trying to predict the risks of marine systems by looking at the invasibility of those ecosystems. Terrestrial systems, it's all about disturbance or eutrophication. Is that a process that's also driving these patterns you're viewing? Because most of your patterns have been describing pathways, but I think underlying that there must also be some potential changes to the underlying ecosystems at all, or are there pristine ecosystems that are easily invasible?
Yeah, there is quite a bit of work looking at that. In fact, my PhD work was specifically looking at whether disturbance facilitates invasion or whether natural communities are equally invadable. And the answer was yes and yes. So what you did see is disturbance increased or enhanced the opportunity, especially for lateral overgrowth, but intact systems were equally invadable by larval settlement. So it's a really interesting kind of question there. The difficulty, of course, is you're asking questions about disturbance in an environment that essentially is a satellite of another location, right? So Coos Bay, where I did my work, received about 100,000 metric tons every 24 hours of Japanese coastal water. And so the flood of non-native larval inputs occurred much greater and at much greater densities into that system than elsewhere. So it's naturally disturbed. You add on top of that frequent dredging, non-native habitat, coastal hardening, fishing activity, and of course, it's very difficult to differentiate that, Bill. You look at the open coastal environment, and it's a very different story. Very few invaders are able to make it outside of those estuaries, and if they do, they are maintaining a toehold. But I suppose the challenge that I would put to that is, so all of that ballast water is coming from estuaries and moving to estuaries. So if we picked up open coastal species and transported them, yeah, really nice retirement experiment. Could you see a similar rate of invasion? And of course, the challenge is, as you would know, Jim Carlton has been looking at the effect of the Fukushima tsunami event and the transport through flotsam and jetsam of those coastal species to the Pacific Northwest to ask the question, do these things succeed? Is it in a scale of 10? Is it as successful on open coastal environments when it's been drawn from open coastal environments? Still waiting on the answer on that one.
Opportunity for perhaps one last question before we head out for outside.
I was just wondering how the eDNA sampling might work, like where would you do it and how far advanced is that as a technique?
Sorry?
The eDNA?
Oh, so eDNA sampling is being employed quite broadly. It's still, so eDNA is an interesting one. You got the real proponents and you got the, I'm not so sure. eDNA will pick up the ballast water, the material that's in the ballast water that gets dumped into the ocean. It does not demonstrate the presence of a population, but it does give you a hint. And that hint is sufficient for you to investigate further. eDNA is rapid. It gives you a much broader understanding of what's available and what's there. Those 2 million samples that we got from the port surveys took that team of 12 the rest of the time over that 6-year window. And it's very difficult. And, you know, as I'm sure the taxonomist in the room will tell you, it's wearing because we have so few taxonomists across those 35 phyla that you really rely on eDNA to help you with those identifications, but it's only as good as the database. So if we don't actually have in the database the species that we're interested in, it'll give you the next best approximation. So it can give you false information as well. I think it's fantastic technology and really want to see it work in parallel. Some of that trans-Tasman sharing is sharing DNA samples for things that each other does not have, so that we can actually develop that database much more significantly.
James, I know you had a question, last question for you.
It's not possibly just been answered, but I was wondering about the lack of taxonomists, and is there potential to replace them with AI?
Yeah, the answer is no. You know, I'm sure that eventually lecturers will be replaced by AI, right? It depends on what you want. And again, AI is only good as the training and the data set behind it. But I think the nuance of technology, taxonomy is what you need to think about. For many of these species, we get partial specimens, we get larval specimens, and for many species, you know, take the jellyfish. You got a benthic form, you got a planktonic form, and we know both of those, but we don't necessarily know that they're the same. DNA will help us with that, but the identification becomes problematic because of the morphological changes that occur through time. So I think it's a long way off before AI replaces the taxonomists. Maybe for spiders. I mean, that's probably a really good one. But it's a very difficult task. I think I was really interested in whether AI could help with gut information around polychaete consumption. So if you can retrospectively identify based on the spicules which polychaete was eaten, yeah, that would be a good use of AI.
Chad, thank you for an outstanding presentation and that journey through marine biosecurity, both here in New Zealand and Australia and places. A lot of rich information about these things. My reflection is that there's one you put up there. I think it's the mantis shrimp, which is the copra harder. They make the most magnificent snapper bait if you ever manage to catch them otherwise.
And they're non-native, so you don't have to feel guilty about using them.
But there's probably too many of them. So thank you for your time today. It's absolutely wonderful.
Thank you.
And the boy for some drinks and refreshments. Thank you.